Hello. Yes, my name is Lucy Davison. It's a real privilege to be asked to speak at this session. I'm really excited about being part of this whole endeavour. I come to this as a real newcomer. I'm a veterinary clinician with a background in diabetes research and endocrinology. But actually over the last 18 months, it's been a pleasure to assemble the mascot group, which stands for Mapping Animal Susceptibility to Coronaviruses, Outcomes and Transcriptomics, thanks to some UKRI funding that we were able to gain last year. And I'm really representing that group here today. I'm sorry, I managed to mute myself there. So the group are listed here. Those in purple and blue have directly contributed to the data I'm going to show you today, but there's actually a large number of people who participate in our regular meetings and bring all sorts of perspectives to our studies of companion animal coronaviruses. So we have Tom Heron and Marsha Wallace's bioinformatics. We have Sarah Falcone, who does a lot of the practical stuff in the lab. And actually, we've really benefited from the foresight of Judy Mitchell in terms of collecting and archiving canine samples over the last 10 years or more. And similarly, Emily Barker at Bristol, who's done the same for feline samples. Importantly, we do also have human medics in our group. We have expertise on MHC genetics. We have a veterinary pathologist and various other inputs from different places that allow us to try and draw in experts from different areas to move our project forward as effectively as possible. And I'm sorry, I'm having trouble moving my slides on. There we go. And actually this work, strangely enough, arose out of a number of us in this partnership already working together on a canine diabetes genetics project. And in fact, we started about 4 or 5 years ago using things like whole genome sequencing, transcriptomics, to try to dissect the genetic basis of canine diabetes and susceptibility of different dog breeds to diabetes. And when the pandemic came along, we decided that it would be a good idea and useful to apply some of our bioinformatic pipelines and our data analysis and our tools to looking at susceptibility to coronaviruses. And we have 2 projects running in parallel. I'm sure many of you are aware of both canine respiratory coronavirus and also feline infectious peritonitis. Canine respiratory coronavirus is something that's been around for a few decades, but actually only really recently have we started to understand what goes on, particularly in places like boarding kennels and also rehoming shelters, where any dog who arrives without having been exposed to the virus usually seroconverts within 3 weeks. It's highly contagious and it's thought of as a type of kennel cough. But actually, critically for this project, for over the last 20 years or so, the RVC have been archiving tissue samples from dogs from various different locations with knowledge of whether or not they have that virus. And most dogs recover very well, but some dogs go on to develop fatal pneumonia. And we don't really know why that happens in some dogs and not others. So we've been using whole genome sequencing and MHC analysis to try to dissect this genetic basis of differential outcomes. And we're now at the point of narrowing down those variants that we think are involved. But we've also had a unique opportunity to go back to archive lung tissues with and without coronavirus and with and without lung pathology present in association with coronavirus, and start looking at what genes are transcribed to try to understand what's different about those dogs who apparently brush off coronavirus with no problems versus those who go on to require euthanasia because they've got extremely severe pneumonia. And our plan and our hope is that in doing this, and this is, I think, what we pitched to the UKRI, is that not only will we find hopefully new understanding of genetic factors, susceptibility to the really severe forms of coronavirus-related disease, maybe treatment targets. We'll also understand more about how companion animals respond to coronaviruses, which could end up being important looking ahead. And especially with the CAT project, we're also interested about delayed COVID complications or delayed coronavirus complications. And in our CAT project, we're looking at feline infectious peritonitis, which is caused by feline enteric coronavirus, and it's an alpha coronavirus, but it does share some sequence with SARS-CoV-1, and it causes a highly contagious diarrhea syndrome, particularly where cats are kept close together in catteries. So of course we do see it a lot in terms of our clinical practice. But what we do see in a small number of cats who've had a historic coronavirus infection is that months to years later, they develop a granulomatous or an effusive disease with an aberrant immune response and hypergammaglobulinemia, and typically that's fatal, although people are starting to treat that now with various antivirals, but it has over the last few decades always been a fatal disease to cats. And it's a delayed complication of a coronavirus infection that we really don't understand why it happens, other than we know that there's a mutation within the host and it allows the virus to change its tropism from epithelial to immune cells, and that's how it manages to become more systemic. And again, we've taken advantage of a really fantastic foresight of people at Bristol and Cambridge in archiving tissue samples from affected cats. So we've been able to do something similar. We've been able to do whole genome sequencing of cats with and without FIP. We have discovered, and this is part of our plan to try to improve resources for people studying companion animals, the feline genome is nowhere near as well annotated as the canine genome. So we found ourselves, particularly in looking at the MHC, and actually having to do our own genome assembly using a long read sequencing technology, because the feline MHC just isn't that well covered. And we're in the process now of taking forward our targeted sequencing to try to home in on those variants we think might be driving certain cats towards this delayed fatal complication of an early life coronavirus infection. And we've also been able to take RNA sequencing data from liver, lymph nodes and lungs from cats with and without FIP, and again use our pipelines developed in the diabetes work to understand what's going on there in the same way as we have done for the dogs. And we hope that we're going to be able to make a contribution to understanding not just companion animal coronavirus infections, but also with human medics in our group, we have an opportunity to have quick and ready access to the right people to talk to about whether or not this is going to be relevant to human coronaviruses. So just to give you a little bit of an outline of the sort of data we're generating, we're particularly interested in those dogs who have coronavirus but are not showing any clinical signs in their lungs or actually clinically. And what we've been able to do is pull out the differentially expressed genes in the lungs of those dogs, and we can see that actually there are certain immune response genes that appear to be critically important in meaning that those dogs don't actually have a particularly severe form of disease. And those dogs who have more dramatic lung signs associated with their coronavirus infection, there's a different set of immune response genes in those dogs. So by pulling those apart, we might be able to think about new therapeutic targets. Similarly, in the cats, we can see that we have nice transcriptomic profiles from all the different tissues that we've looked at. It's a little bit more complex in the cats because not every tissue in the body is infected, but certainly we see in the liver there are more than 600 differentially expressed genes in FIP, and in the mediastinal lymph nodes we see more than 300 differentially expressed genes. And digging into these genetic profiles, we're able to look at the importance of the type 1 interferons, gamma interferon, and various other antiviral genes. The other thing, as I mentioned, is we've put together a new feline genome assembly. You can see on this figure on the right-hand side, on the top assembly, it's not very contiguous in that area that's highlighted in pale orange, and we've managed to improve the feline MHC genome assembly, which we hope will also help us understand some of the susceptibility to more broadly different types of infectious diseases in cats, because there's a big gap actually in that at the moment. So we look forward to sharing that data with the scientific community. So we're waiting for results to come through. As I said, we're very early days in our exploration of infectious diseases, and we've come to this from a completely different place. We're looking to try to pull out viral reads from archived samples as well. We're looking at the relationship between different pathogens in canine lungs and serology versus genetic data. And also there's a re-emergence and interest in vaccinating dogs for canine respiratory coronavirus. And in cats, actually, the other thing we're starting to think about doing, as well as what we're doing in dogs, is thinking about the immune cell repertoire analysis in the lesions themselves in FIP, because we know that part of the problem is actually the immune response to the virus, and cats give us a really fantastic opportunity to explore that in more detail. So it just remains for me to say thank you very much for your attention. I'm very happy if anyone wants to ask questions, or just drop me an email at either my Oxford or RVC email address. And thank you again for making me part of this. It's a really exciting and interesting group to belong to.